So Justine and Dan are both part of the Canola Council agronomy team. I just want to take a note, and hopefully Angela and Warren can still hear me, that this is an extremely valuable team for us. We, as the Manitoba canola growers, do not have agronomists on staff, and we lean on the council team. It's a real strong partnership, and our members benefit from that. So thank you all for your support. So Justine covers off Western Manitoba, Southeastern Saskatchewan as an agronomy specialist. She leads in the blackleg management topic and chairs the Canadian Blackleg Steering Group. And Justine is also a sucker for punishment and doing her part-time master's at the university while working as an agronomist. And Dan joined the council in March 2011. Dan's scouting led to the first detection of Clubroot in Alberta in 2003, so I think we all generally blame Dan for Clubroot, but he's got thick skin, he's all right. Since that, he's taken on the role of Clubroot leader in the agronomy team and is on the Clubroot steering group. Clubroot steering group, and he covers the north central Alberta region, so definitely first-hand experience of the extremes of Clubroot. So appreciate them joining us this morning to give us a little more insight. Thanks.
Thank you.
I'm just going to grab a water too.
Here's your clicker.
Is it there? Okay.
Awesome. Good morning, everyone. Thanks for joining us here this morning on the FCC Theatre. Delaney already introduced me. I'm Justine, and this is Dan, if you haven't figured that out. Today we are here to talk on Clubroot. With any luck, this clicker is going to work magically for me. Maybe, maybe not. But we want to send a huge thank you to Manitoba canola growers for having us here today on the stage. We worked side by side with them on a lot of agronomy extension components, so it's always grateful to have that opportunity to speak to you guys here this morning. So with that, we are going to talk on Clubroot management. We've got a different approach to it. Hopefully you guys like it. And we're going to try to throw some guns and some excitement into here, even though Clubroot's already a pretty exciting topic. Just what we're going to cover today, so you're aware of what we're going to go through. We are going to highlight Clubroot, just the background, the basics, so what this disease is like, where it's at in the prairies, and how bad of an issue is it actually, and then look at some management practices. These are going to be called our silver bullets for now, but you'll see that they obviously don't work by themselves. So that's going to be that overall shotgun approach, which we are going to kind of summarize with the importance behind using all of these tools together. Key points, because we've got an hour with you guys, we're going to go through quite a bit of information. But while we're going through this, if you can kind of remember these three key things as we go through it. Number one, keeping spore loads low. That's going to be a reoccurring theme that you see with a lot of the material that we're presenting. Second being that resistance is futile. We cannot rely on resistance genes or genetics alone to manage our problems, and we're seeing them fail time and time again. We've got really, really great products there and available, so we want to be able to extend that longevity of those resistant varieties. And then overall, just adopting this integrated pest management approach. This is going to be our shotgun approach that you're going to see throughout. With that, I will now hand it over to Dan. Maybe.
Okay, thank you, Justine. So this slide here shows the life cycle of Clubroot, and if we start in the top left-hand corner, you can see the resting spore. This is what overwinters in the soil, and when a crop is planted, the spores sense that there's a plant there, and they come out of dormancy, and they can actually swim a short distance, and they find the root hair. At that time, they infect the root, and a not very well understood process happens, and a secondary infection comes along, and that's what forms these giant clubs on the roots, and that keeps the plant from uptaking water and nutrients, so it will wilt and eventually die. And then the whole cycle starts over again with millions and millions of spores being put back into the soil to go back into dormancy, awaiting another crop to come along to infect. So our resistance works in between the primary and secondary infection. That's where our clubroot resistant varieties work. That's why they don't get clubs if they're truly resistant to the strains that we have in the field.
Yeah. So the Latin species, Plasmodium brassicae, that's something that you'll see in a lot of publications. If you're not aware, that is clubroot. That is the primary species we are dealing with here. Because I work on blackleg all the time, I want to make the emphasis that clubroot is a soil-borne disease. So anytime that we're working with any sort of soil, you have the potential to be moving these spores. It's not like other diseases like blackleg that needs that old residue, that old canola stubble to house itself on. So that would be a stubble-borne disease. So that's one of the key differences, and that really changes your management techniques when you're trying to deal with a disease like this. Ooh. Getting trigger happy here.
There. So this is an awesome slide. It's a highly magnified, obviously. Each one of these is a plant cell, and you can see the cells are meant to be hollow to transport water and nutrients, and they're completely plugged with clubroot spores here. You can see hundreds and thousands of them in one single cell. So that's how tiny these spores are. And I think this next slide emphasizes really how small they are. They're about the size of a red blood cell. So obviously you can't see them. You couldn't even see 1,000 of them if they were together, I wouldn't think. So they're very tiny. I think something like 10 of them can fit across the width of a human hair. So again, they're just so tiny.
Yeah. So when we're looking at the overall life cycle component, clubroot, right, our only real evident piece is seeing the galls forming on plants. We're not seeing spores in the soil. As much as that previous graphic is pretty neat, you're never going to see that with the naked eye, right? So this is how clubroot can go undetected for so long. It's so tiny, and there's so many spores hidden away in that soil.
I would think that also is what makes it so difficult to control, right? Because even the smallest amount of soil or dust blowing around could easily contain thousands or millions of spores, right? So a lot of times it is in your control if you move clubroot around, but it can also be out of your control too, right? If it's blowing in the wind, that's hard for you to control.
Go ahead.
So clubroot goes where dirt goes. Like Justine said, it's a soil-borne disease. So whether you're growing canola or peas or barley or wheat, if you've got clubroot in the soil, you're going to move it around. Canola will propagate clubroot, but it doesn't move it around. What moves it around is soil movement, particularly with equipment. We've seen pretty much every mode of transmission in Alberta, from animals to hunters to oil lease sites, power lines, you name it. We've seen clubroot move that way, but primarily it's moved by farm equipment, moving from field to field, and we do see it at the entrance most of the time in Alberta. Probably 90% of the fields that have clubroot, it's at the entrance because it's been brought in from another field that's been contaminated and the equipment isn't getting cleaned.
Yeah. Manitoba's a little bit of a different situation. A lot of the new cases that we're finding here within the province have been coming in through waterways or runs, so not necessarily at that field entrance. When we talk about scouting here, which we will shortly, the recommendation was to go into your field and go whichever direction equipment was going to be dropped at first and look there for spores, but that's not necessarily the case here in Manitoba. We are finding a lot of it coming through waterways or other components, but still moving within that soil.
This is a little bit of an older slide, but the yellow square in the center around Edmonton is depicting where the wind currents started when they were tracking reverse trajectory wind currents. And the dots that you see scattered across the prairies are how many times wind currents started in Edmonton and ended up somewhere else. So you can see, I think it's Portage, there was one event over there, and Dauphin had 2 events where soil particles were tracked from the Edmonton area. So I would say undoubtedly they would have drugged some clubroot with them. So again, it's not going to always be in your control whether or not you do get clubroot. It could be just blowing in the wind. So that's why this shotgun approach of doing everything you can to keep it from becoming a problem on your on your farm is very important.
Yeah, this is also just an indicator. It's a matter of time before we all have Clubroot, so there's no shame in that. It is spreading. Every year we find more and more fields, and we'll highlight the maps here right away. But we are seeing that right through wind events, it is able to spread across the prairies. So you might be okay right now, but if that perfect wind comes across the prairies, comes across Saskatchewan, you might be at risk, right? The spores do travel via wind as well within the soil. So.
This slide is Mary Ruth McDonald has created it, so it's not exact and to the exact spore number, but typically in the past, until you get up to around 100,000 spores in a gram of soil, which sounds like a lot, but it isn't. Until you get to that level, you tend not to see symptoms in the plants. You have to have really nice, warm, wet conditions for lower spore loads, like 10,000 spores per gram or even 30,000. Before they're going to show symptoms, you need to have really conducive weather too. So in Alberta, we're at millions and sometimes even a billion spores in a gram of soil. Believe it or not, there's room for that many spores and still soil in there. Out here and in most of Saskatchewan, you would be way, way lower than that and still have the opportunity to keep it that low. So you might be at 10,000 spores per gram and you won't see any symptoms above ground. The plants won't be wilting. You would never ever know that clubroot's there unless you pull plants out of the ground and look at the roots. And you'll start to see, like you see at the 10,000 or even at the 1,000 level, you can see on the side roots there's just some slight swelling. And that's the start of clubroot. If you catch it this early, that's incredibly awesome. We didn't really catch it in Alberta till we were at the millions and seen massive dead patches appear. And by then, we're kind of battling like a dog chasing his tail at that point. So I highly encourage you to go scouting, and I think you'll start to find it at these low levels. Much earlier than waiting till you can see it driving down the road from a dead patch appearing.
Yeah. One key piece with this is also it only takes one spore to cause infection, but because we're out in the environment, it becomes a numbers game. So that's why we're seeing real big galls starting to form with that million spores per gram, because you've got such a high amount of inoculum there. When we look through the maps, you are going to see that some of the maps are presented based on soil samples and how many spores per gram of soil. So this is just something to kind of keep in the background where you are going to start seeing those symptoms. A lot of the fields here, or I shouldn't say a lot, some of the fields here in Manitoba that have been found with clubroot, they are kind of in this range. You're not seeing that necessarily dead patch in the field. The plants looked good, looked healthy. You wouldn't have known that you had clubroot and all unless you were pulling up and actually looking at that root to see the galls starting to form. Some of the real severe situations, which we have found as well in Manitoba, are showing the real big galls already starting to form. Our neighbors to the south in North Dakota also have many fields that look like this as well. So there's talking right into that levels of millions of spores per gram of soil.
Yeah, and at that point, you certainly can see above ground symptoms, but yeah. I think everybody is shocked in Alberta when they first see clubroot, just how bad it is below ground before you see symptoms above ground. Clubs the size of your fist below ground, and it's very hard to tell anything is wrong at all with the crop above ground. It might be slightly thinner or maybe prematurely ripening by a week or two, but nothing really, really obvious appears above ground. So it's so important to keep pulling plants even if you don't think anything's wrong. Yeah.
And with the issue when Dan was going through the life cycles, right now when this gall starts to disintegrate, it's going to release all of its spores back into the soil, right? So it's keeping that level of spores in the soil quite high when you've got galls that large. That's all you.
Okay, so this is the timeline really of clubroot. It was in vegetable crops all across Canada for years and years. It's an old disease, but it's new to canola, I guess. And in 2003, a handful of fields were found north of Edmonton. And then slowly it started to spread and be found more. I think the more we look, the more we find it, really, to be honest. Even in Alberta, some of the outlying areas that don't think they have clubroot do, almost guaranteed. They just don't have the survey and they don't have people looking for it. So we started looking for it more. In 2008, it was ID'd in Saskatchewan. 2013 is when we seen the first resistant variety not work so well anymore. 2015, there was 10 pathotypes identified. And believe it or not, now, as of this year, there's 36 different pathotypes or strains of clubroot, of which many of them are able to overcome resistant varieties. So this is the big problem in Alberta, is we didn't take a proactive approach, and now we have the resistant varieties not working for us anymore, and that's a really, really big problem. I think there's probably been a couple dozen fields now that I've been to where they just can't grow canola anymore because we don't have genetics that work on the strains that they've kind of bred for in their field by simply growing too much canola and letting the spore loads get too high.
Yep.
And there's a worldwide distribution of clubroot. It's all over the world, and it is a little bit overlaying with soil type and moisture and pH. If you put them all together, they're somewhat related, but the biggest factors by far and away are moisture and heat. Those are the two things that get clubroot rolling, and unfortunately in Manitoba, you have a lot of moisture and heat, so I think that your environment is very conducive for clubroot here as well. Yeah, we seem to really like our plant diseases here just because of the environment. So unfortunate for us, we have to deal with them all. One thing to point out on this map too is this is worldwide, and this is for brassicae, right? So this is not just canola production, and that's kind of the one key thing with clubroot. It's not just a canola issue. It is found in a lot of these vegetable crops, which Dan had alluded to. So there's lots of information to gain from. I know we're learning lots from the greenhouse industry and how they manage it, and that's how we've been slowly adopting some of those management practices to then use here in the Canadian landscape. Here's a Climax annual growth index. So really what it is a predictor of how could clubroot spread. And I love this slide. If you look kind of in the middle left side of the graph, that's kind of where we're at right now. And if we get more rainfall and more heat, you can see that pink, kind of whitish pink region expands to a larger area. And in the top right-hand corner is 3 degrees warmer and 20% more rainfall. And you can see pretty much all of Manitoba has turned to that whitish pink color, which would indicate that conditions would be perfect for clubroot to get established very, very quickly here, just as quickly or quicker than it did in central Alberta. So it's just a kind of a prediction model, and in summary, it shows that Manitoba is very high risk.
Okay, we're going to show the maps and where we are finding clubroot currently. We've put them all on one here for right now just to explain the differences. These graphs are presented differently across the provinces, right? They're done by the provincial groups or by universities, so they're not necessarily working together, so you do have differences. Just starting off in Alberta, right, that being the hot zone where clubroot was first really established in the prairies, the way they present their map is based on actual plant symptoms, so finding gulls on those plants. They don't do a soil test. It'd be neat to have a look at that. I'm sure that our map would change entirely to red. There would be so many spores across it, but that's not how they present it. They present it on a percentage of fields, or the fields surveyed, how many are actually showing gulls. When we move into the Saskatchewan map, Saskatchewan for the longest time just did not survey. If you shall not look, you shall not find. That was their approach. Thankfully, the last 2 years, they've done some extensive surveying, and they have found Clubroot across their northern territory. The way that they work, they've got it on a color scheme, looking at the fields. So orange being that there was over 10 fields within that county showing or actual galls or plant symptoms. They also do soil tests. So on the map, you are going to see little patches of blue where they found spores within the soil, but no plant symptoms. That then moves into our Manitoba map, which I know that's primarily what the audience here is interested in, and it's based on visual symptoms and the actual soils per gram, or a spores per gram of soil as well. So you're going to have both of them combined on that Manitoba map. And here is the map. This is the 2019 provincial map done by Manitoba Agriculture. A few years back, I believe it was 2014 or 15, they did a really extensive grid survey of the province, and that's where they took soil samples and were able to detect these different levels. So that's why our map is so colorful. The green is showing that within that RM, the fields that they were surveying, they didn't find any plant symptoms, but they were finding spores at a really low level. So the scale on here is 1,000 spores per gram of soil, so at a really, really low level. And if we think back a few slides to the visual symptoms, right, we weren't seeing any galls starting to form at that 1,000 level. Manitoba now, up to 2019, we've got, I believe it's 8 RMs that have confirmed plant symptoms, right? So actual showing galls, those are in the orange. So figure out where you live, right? We've got this hot zone here down in the center of Manitoba. Swan River Valley also has confirmed symptoms. And the one new RM this year that was found was actually located in the Interlake region. Right, so separated off from that primary canola production area, not around where these other fields have been found. And this is a case where it was coming in through the water, right, coming off the lake system, the river system. So I believe we're up over, gosh, I think 38 fields, 36, 30, yeah, it's in that range of actual confirmed fields with Clubroot. And this number continues to grow each year. We've mentioned how Clubroot likes that warm, wet conditions. We've actually found a lot of fields the last 2 years because it's turned dry during the summertime. So those patches in the field have become much more obvious. Myself surveying in the south here, looking at some of these fields, there was no actual above ground symptoms, right? The plants looked good, they looked healthy. Just at the end when you're pulling them up is when I was seeing galls. But yeah, the last 2 years with the dry conditions, it's made those patches a little more evident because that constriction is happening at the roots with no moisture going into those plants. Manitoba also presents a risk graph, which is, I realize I've got a few years later, but I know this hasn't changed a whole lot, but this is based on the percentage of canola acres grown. So when you compare the two maps together, you can see where those hot zones are. The real dark red in the acres is over 50% of the acres are in that RM are growing canola, right? So that's just that increased intensity of canola production, right? We've got Clubroot up in that area. We've got Clubroot down in the south as well. Those are areas of intense production of canola, right? They overlap very hand in hand. This is the Saskatchewan map. So this is updated from 2019 in their surveying efforts. They haven't done a lot of surveying in the south, as you can see, but their primary production of canola is kind of north of highway, or of the Yellowhead Highway, or Highway 16, right? So north of Yorkton up into Saskatoon. You can see the northern half has Clubroot all throughout. If we jump across these RMs right across from Swan River are also showing symptoms, makes sense. Any of the blue patches on this map are just that the spores were tested, or the field samples were showing spores. No actual plant symptoms were found yet. We've got one RM that's got over 10 fields. So this is just because Saskatchewan done a lot of surveying. They're starting to increase that number, so they're starting to gain more knowledge and information of the actual risk and what situation they are at. But each year that they survey fields, more and more are turning into Clubroot cases. Alberta. So Alberta just recently has changed their map. Before it used to be the real colorful map. You saw that really big red zone around Edmonton area. They had to change their scale because I think the low, what was the high, or the high number was over 50, yeah, over 50 fields per RM or per county there. They're now finding over 1,000 fields per county. And that's just because they've been surveying for close to 15 years now, over 15 years. So right, the number of fields that they're finding just keeps on growing. One thing to really point out with this map is, right, it is just based on actual physical plant symptoms, so seeing the galls. There's no soil samples included in this map. Though only one county is highlighted in red with that over 1,000 fields, that's the Leduc County, and that's because they do a really good job of surveying. So their ag fieldmen go and they do survey most of their canola fields there, and they're able to just find it. So they've done a really good job there. So I don't want to single them out as a real bad spot. They just do a really good job of scouting. All the other RMs around Edmonton should be red as well. It's just coming into actual going and finding those symptoms. Anything you want to add to the Alberta?
Yeah, I think it's important to understand that soil sampling is a tool in the toolbox, but it's not a really reliable tool. Excuse me. We've had fields in Alberta where we pull up plants that have massive galls and the plants are dying, and we soil sample and it comes back negative. So that's disheartening. But we've also tested fields where we get 1 million spores in a gram of soil right here, and we get 0 right here, and we get 1,000 right there. And so if you take 12 cores randomly in your field and mix them all together, that number doesn't really mean anything, right? It could be mixing a real hot spot with 10 cores that are 0, and it would make you look like you don't really have clubroot. It's so patchy in the soil, and the spores are so tiny that it's not a super reliable technique. I think the researchers around the world would tell you that the number one best way to indicate if you have clubroot is plants, pulling plants. Number 2 would probably be a DNA test that you would send away with a plant and the roots. And number 3 would be soil testing. So it is a tool, and if you come back in the millions in your soil test, you know you have a problem. But if it comes back extremely low or even at 0, it easily could still be clubroot there somewhere on your field, and it could be kind of pretty bad too. So Justine's right that in Alberta we see more often at the entrance, and in Saskatchewan and even in Manitoba, it seems to be a little more in water runs and low spots. And to me, that means that it's been brought here quite some time ago. It's just that those are areas that it likes. It loves the low areas. It doesn't fly around and look for low areas. It's just that that's where it prefers, and that's where it can propagate easier. So I would suspect it's often at your entrances too, but they're just not as conducive to show symptoms as your water runs would be, and areas with more moisture and things like that. Yeah. And we are seeing in Alberta, when we get new strains that are overcoming resistance, typically they are in the hot spots like you're finding it in Manitoba. They're in our low areas, or our compacted areas that remain flooded, or the stressed out areas. Just because that's a better environment for clubroot to live in, I think. Yeah.
See if that's going to change. There we go. So now that you've got a background of clubroot and what it does and where it's at across the prairies, this is where we're going to kind of get into the meat of the presentation. And we're going to talk about what we've titled as silver bullets. I'm sure you guys have seen this presented in other ways. We've also called it our clubroot recipe, right? You need all of these ingredients involved. But what Dan and I are going to do now is we're going to go through each of these 6 best management practices, bullets, these silver bullets, and go through why they're important, but why they don't stand up by themselves, why they need to be incorporated with other techniques, and we can't rely on just one of them. So we will show this slide again, but we are going to work through each of these step by step here. This is where my clicker is going to get real mad. Okay, so number 1 we're going to talk on is scouting. Click. So the easiest thing, and Dan and I have already said it a few times, when we're scouting for clubroot, you go into the field and you have to pull up plants. You physically have to boots in the field and pull up a plant and look at its root. And not just one plant, several plants throughout that field, right? Our recommendation originally was at that field entrance, move to the low areas, the high traffic areas. If you have Manitoba Hydro going through, if you've got oil and gas, right? Any area where you're going to have that risk of lots of soil transfer or movement. Go and pull up plants there, canola plants at that time, and look. Even if you're growing a resistant variety, we are going to touch on resistant varieties here in a couple of slides, but even if you're going clubroot resistance, you need to go and pull up those plants and see how your genetics are performing. The earlier we can manage, the better off, right? If you know you've got an issue with it, then you can actually do something about it. And we do a really poor job of scouting overall as an industry. We've let these cases of clubroot grow right out of hand. I'm going to flip to the next symptoms. There's a lot of misdiagnosing and confusing. Clubroot's a pretty obvious one to figure out if you would actually go and pull up plants. Some of the symptoms here, we've got, this is a young canola root, you can see, and it's just got tiny, tiny little swellings on it. So that's going to be probably something you would see in a situation where you've got lower spore loads. This is the big nasty one. If you're finding plants that look like that, clubroot has been there a very long time and has been established. This one is just a prime indicator that we've done a poor job of scouting. And then at the end, this is the top image. That's what clubroot galls look like once they've disintegrated. So at the fall or after harvest, you go and pull up a few plants. Maybe you're doing your plant counts. This is also a good time to look. You will see the decaying galls, and they're going to smell and be disintegrating, and kind of just brown mush is what it looks like. Very obvious what it is if you would actually go in and pull up plants. Anything you want to?
Yeah, I think it's a good point that Justine makes, that they're giant galls and they're easy to identify as clubroot, but not above ground. Even that middle plant that you see with those giant galls, that crop was still green. I've been to many crops that you wouldn't believe the size of the galls, and the farmer doesn't really know what's wrong yet because they haven't pulled plants out, but they know that something looks strange. You know, we've been to patches that are 30 acres in size, and when we get there, all the plants look like that middle plant that Justine showed. In a 30-acre patch, and really all it was prematurely ripening, maybe by a week or something. It looked like a low area, so I think you just need to pull.
Yeah. If you have a patch like this, we always talk with clubroot, typically a patch area. Once again, this has been there and established for an extremely long time. This is an indicator of very poor scouting, because someone should have been noticing this in years prior. But this is what you'd be looking for when scouting for severe cases of clubroot. Something on a dry year, right? You're going to have those plants die off early, right? They're not getting that moisture into them. We are going to see patches like this. We've talked lots about the premature ripening. This would be another example. This is a very large, obvious patch. Something like this should trigger, I should go look at those plants, pull them up, assess. It could also be black, like it could be something else, but you need to actually go and pull those roots up and have a look. Okay, so that's scouting. So we do a pretty poor job of it overall. So that's one of the bullets that we just cannot rely on alone. Scouting is a proactive measure, right? You need to be doing it before you have an issue, and then you actually find out you do have an issue. That's where you can then apply a bunch of these other tactics. So it is a very important piece, but it's not a standalone. You can't just rely on scouting to solve your clubroot issues.
You know, in the vegetable industries, in other countries and in Ontario, they farm 10 acres and 15 acres. That's their whole farm, and they transplant things, and they can identify and find clubroot really, really early when a patch the size of this table is formed, because they've walked every acre of their farm. With 22 million acres of canola, there's just absolutely no way that we're going to find these small patches from the swather and the combine. And that's an issue, is we don't tend to realize we have clubroot till it's really quite bad, because we just farm so many acres we can't look at every corner of our farm. So that's why this recipe is a bit of a proactive approach that I think the folks in Manitoba for sure can use all these steps to never get to the point where we got to in Alberta. We didn't have resistant varieties. We didn't understand rotation. There was a lot of things that we did wrong and that we didn't have access to, including information that kind of led to the problem in Alberta that's kind of hard to manage right now.
Yeah. Okay, so our second bullet is crop rotation. Crop rotation, you see it in the media quite often. We talk about it a lot. We know for disease management it is so extremely important. But once again, it alone will not help solve our clubroot issues. If we're planning to not grow canola for upwards of 20 years, that's not really in our back pocket right now. So crop rotation continues to be important, but it needs to be incorporated with other tactics as well. Just to show if this is, there we go. This is a really neat graphic. I quite like this. It's a clubroot spore survival, or we're looking at the survivability. So there's different survivability types. Us humans, we are type 1. So, right, we have a pretty decent lifespan, right? We've got our time at the bottom and then the percentage of organisms living. So, right, when we start off with however many kids born, we last a long time. Our lives go well. We hit I'm trying to think what our average survivability is now. I think women's like 87 years or something like that. Right, once we start hitting that 80 part, we start to then trickle off and die pretty quick, which is an awful thing to think about, but that's, a survivability type. Next is our songbirds. So you start off with a decent amount of songbirds. They slowly just die off over time. Like they're pretty negative linear trend. Clubroot spores, or frogs is the example that they usually give with a type 3. When you think tadpoles, when a frog reproduces, it produces hundreds of tadpoles, but they die off really quick. Very few tadpoles end up surviving to become that frog or that toad at the end. Right? So we start off with lots of them. They die off really, really quick, and there's only a few that can make it, quite a few years into the future. And that's what happens with clubroot spores. Right? We're talking millions of spores per gram of soil, or millions of spores being released from one gull. They'll last within the soil for maybe a few months, maybe a year. Even within that upwards of a 2-year period, you've lost most of those spores. However, you are going to have this population of resting spores in the soil, and they're going to stay in that soil, and they're going to stay dormant for a really, really long time. And I think we're upwards of 20 years of work where we're still finding resting spores that are hanging out. Do you have anything to?
Yeah, no, that's right, and that is again something we didn't realize in Alberta. We were told that all of the spores last for 12 or 15 years, and you have to be 20 years without canola before you can get rid of clubroot. And there's been a lot of research, particularly in Alberta recently, that shows that within a year or two, so that 2-year break, up to 99% of the spores will die. It's when you don't give a 2-year break and you only have a 1-year break. And we were all wheat canola, so we only were given it a 1-year break. And most of the spores were still hanging around. Had we known that 1 extra year out of canola and going on a 3-year rotation was so valuable, I think in hindsight, more farmers would have gravitated toward this 3-year rotation, but we didn't know that. We were told that you have to be many years out of canola, and that wasn't feasible for them, so they carried on a 2-year rotation. And I think this is the most important thing of the whole talk, is a 2-year break from canola, and up to 99% of your spores will be gone. Unfortunately, in Alberta, when we have 100 million spores, 1% of 100 million is still a million, so we still have a problem after our 2-year break. But that wouldn't be the case out here. So I think that's the biggest take-home message, is that 2-year break is unbelievably valuable.
Yeah. So here in Manitoba, thankfully, we've got cropping options, especially here in the south with the adoption of more soybean acres. I know after 2 dry years, we don't want to think about soybeans, but here's for hoping for rain this next year, which hopefully Drew will tell us in the next presentation that's going to be the case. But, right, we've got that option to go wheat, soybeans into canola, right, to give it that 2-year break. So I know there's lots of misdialogue on that, but yeah, it's 2 different crops before your next canola crop, right? So canola 1 in 3 years really will help reduce the amount of spores for clubroot and for other diseases as well. So I know there's lots of fighting and lots of bickering around what the perfect crop rotation is, but we've got more work now coming out of Alberta within these clubroot hot zones showing that the 2-year break will get rid of, I think it's upwards of 90% of those spores, right? So you are able to really reduce your risk to clubroot if growing canola on an extended rotation.
Yeah, so this slide just shows that the research is quite robust and evident that that 2-year break is the biggest bang for your buck. Certainly there's situations where a 2-year break is nowhere near long enough. You know, when we see spore loads as high as they can get in Alberta, we've been 7 and 8 years without growing canola and still have problems when the spore load's that high. But the biggest bang for your buck is that 2-year break.
Yeah. Yeah, so once again, we can't rely strictly on crop rotation because we do have that risk of spores staying within the soil, but it's just another one of those tools that we need to be adopting with our scouting, and then hopefully our next bullet will here. This is to highlight our Manitoba situation. So we talk about how important crop rotation is. Unfortunately, we don't follow that all that well. These are cropping frequency maps from 2009 to 2014. I don't have new ones, but the new ones would probably be redder because our canola acres have only gone up since then. So I'll keep throwing these old ones up. If you're familiar with the map, here's Riding Mountain National Park. Here's the Duck Mountains, Swan River region. So this is the blow up of the Swan River Valley, or north of the valley. Here's ducks on the zoom up. And how these frequency maps work within that 6-year period of time, when the darker in color you get, the more canola that's being grown on that same piece of land. So yes, this is the government spying on you. They know what you're growing from satellites. They're able to tell that. But we've got a bunch of red dots here. So what that is telling me is that canola has been growing back to back, right? They're growing canola 4 out of the 6 years on pieces of land. So you can see all the red dots. There's even blue and purple. So out of that 6-year period, there was canola growing on that same piece of land for 6 years straight. That is not going to help solve our clubroot issues at all. So I'm picking on that north area right now because I know our options are a little bit limited, but we are seeing the same thing. If this next slide. Here's down at Holland, so same time period. I was thinking, oh, this is great. You know, we've got lots of greens and yellows. Okay, that's canola hitting that perfect rotation, or that ideal rotation, I won't say perfect, of growing canola 1 out of 3 years. But I'm starting to see still a bunch of orange and red. So we still have canola, or that canola wheat rotation happening here, which isn't allowing enough time for those spores to break down within the soil. So we don't do a good job of our crop rotation. We talk how important it is, and we would like to see breaks, but it doesn't happen out in the field per se. So something like Clubroot, it's going to be very tough to manage on those fields that are continuous canola. So that brings us to our third bullet here, resistant varieties. Resistant varieties are something that we've put a lot of pressure on in the last few years. We figure if we grow them, all of our issues go away. We don't have to worry about all the other tools, right? I've got a resistant variety. Clubroot will not be an issue. And I want to mention how awesome clubroot-resistant varieties are, because they are. They work well when stewarded correctly with other tools. This is a great example out of the Leduc County, so right back in that Alberta hot zone. A father and son knew that they had clubroot on this field, and the father chose to grow a clubroot-resistant variety knowing they had clubroot, and the son chose a susceptible. You can visually see the difference here. The father grew 50-plus bushels. The son was down around 10. So this is a moral of the story here for the younger guys. Listen to your dad every now and then. He might know what he's talking about. Anyway, but this is just to tote how awesome resistant varieties work when stewarded correctly. And thankfully...
And that would be all, on that right-hand side, was all weeds. The canola doesn't regrow because it dies. So that's all weeds now. It's not just the fact that he only got 10 bushels, but he's got so many weeds to deal with in the future. And just, I mean, everything is wrong with that.
So thankfully now we have a bunch of options for clubroot resistance. Every company on the market has got a few tools now. So we're starting to adopt more and more clubroot resistant varieties. When we're thinking of disease management, all of our varieties are resistant to blackleg. That's not the situation with clubroot. Clubroot is something that we've been slowly adopting into more and more of our varieties. So you can see the diversity here. There's a few little asterisks on the chart. That's just to indicate a new type of resistance. There's a lot of argument what you want to call it. For the sake of today, we'll call it second generation. Is that? Yeah, second generation. Just wait for that.
Oh.
Yeah, is that yours? Okay. If that's your truck, you probably should go move it. Okay, asterisk, sorry. So that's what we're calling second generation. So it's something new, and a lot of those varieties are targeted to the hot zone in Alberta, so good luck trying to get your hands on them. They're a bit of a hot commodity right now. But the moral of the story is here, we've got lots of options. Farmers here in Manitoba, give them a try. There's no yield penalty to growing something that's got clubroot resistant. These are top yielding cultivars. They're going to compete with your go-to varieties that you've been sticking with. So do try them. There's no standard labeling system. You see a C thrown up quite often. For the most part, that is meaning a clubroot gene of some sort, or that it is resistant. Anything you want to add? Yeah. So to talk about the resistance breakdown that we're seeing with clubroot, we got to understand what's going on in the field. So we're applying this variety that is supposedly resistant to clubroot. Why are we seeing galls starting to form? Why are we starting to see patches or breakdown? In Alberta, they now report their map of the number of fields that are seeing resistance breakdown. They're documenting that. But what's going on, and this is a nice little graphic to kind of show it. So we've got this variety of clubroot resistant variety that's being grown in the field, and it's going to be resistant to the blue spore. So whatever pathotype blue is, we'll just go with the colors. Blue pathotype, it's resistant to that. So it's not allowing those pathotypes into the plant. However, the other pathotypes that are in the background, so we see the red pathotype hanging out, it is still able to infect. So it is able to enter into that plant and cause galls to form. But it's at a low level, it's there, it's hanging out, it's not really doing a whole lot. If we continue to grow canola in that tighter rotation, allowing those spore levels to increase and stay high, we start to see a shift. So that predominant pathotype in the field becomes the red one. And now that red one's able to attack the plant still, and there's more of them, and it's able to drag along all the other pathotypes with it. And that's something new that we found out with it. This pathogen is so smart, it's taking a bunch of those spores that shouldn't be able to get into that plant, but now they are because the other overcoming ones have entered in, and they've started to cause those galls to form, and they're dragging all their friends along with them. And that's how we see these resistant varieties break down so quickly. This pathogen can adapt and overcome and change within a cropping cycle within the field. So the spores are smart, and they're finding a way to overcome what we're throwing at it.
And there's just so many different strains out there now that are found in Alberta that undoubtedly every field is being put under this pressure, where there's some spores in that field guaranteed that can overcome the resistant varieties. And so the more we grow it, the more those spores will be present.
I'll let you.
This is where the fields are breaking resistance, and we don't do a very good job of scouting in Alberta. Some of the counties do a really good job, and those are the counties that are finding a lot of fields. So I would suspect we're at thousands of fields where the resistance isn't working properly. But we can't look at them all. And they tend to appear in clusters. So someone north of Edmonton will find this new strain in their field, and will look at surrounding fields and find it all in that area, that same strain, from moving soil around still and growing too much canola. But certainly, I think you guys in Manitoba you have found a field or two where these new strains are appearing, but I wouldn't suspect that you would find it very often. It would be very rare, and that's why kind of this proactive approach of doing all these things we're saying will keep that spore load low enough. To me, it's like getting a flu shot. If you do all these steps, you don't get the flu shot after you get the flu. So you don't want to start doing these steps after you get clubroot. I think you need to do all these steps before and treat it like a flu shot.
So we've talked about pathotypes. You've heard that name come up a few times. That's what we call the different strains or types of clubroot spores within the field. I'm just throwing this up. I don't recommend anyone even try to interpret what's going on. It's confusing, it's complex. Because we've got so many resistant breaking pathotypes in Alberta, we've had to create a new differential set, and that's how we determine what pathotypes are what. This set continues to grow. The newest or the latest one is actually 36 columns, but this is how we determine if it's going to be resistant to a variety or not, or to a different species of brassica. So nothing to take away in there, just that it's extremely confusing. You're going going to see your varieties labeled with different pathotypes on the bag. Here in Manitoba, what we need to worry about is just growing a clubroot-resistant variety to start with. The pathotypes, there's a bunch of them, and it keeps changing. We'll leave that for the researchers for probably quite a few more years, because every year this continues to change and continues to become more and more confusing. Yeah. Yeah. I'll skip over that one because that's more pathotypes. We had highlighted that there's over 36 different pathotypes here in Canada.
So in a given field, when they go, when it has clubroot and they test for what different pathotypes or strains are there, there's a lot in every field. So in one of those galls that form on the plant, on the roots, there's many strains in that gall. It's not just one strain. This is the issue, is there's many strains, up to 36, right? So you'll never find a field that just has one strain in it. It will have a population of all sorts of different strains. So when you grow a resistant variety, some of those strains are going to be able to infect the plant still.
Yeah. So we've covered off the big three, like the real crucial ones. The final three that we're going to whip through here quickly are practices that will make a difference and should be adopted, but we know a lot of people don't necessarily do them, especially this minimizing of soil movement. The best way to minimize soil movement is to clean your equipment, which I know does not happen. Right? It's very consumes a lot of time, but all we ask is if people could knock off the big clumps of dirt or stay out of the field when it's extremely wet, right? If it's wet, you're then taking lots of that soil with you and a lot of those spores. If you're able to just knock off the big clumps of dirt, you're already reducing your risk to over 90%, right? You've gotten rid of most of those spores. If you decide to then spray it down, you're upwards of 95, and if you're going to then sanitize equipment with a bleach solution of some sort, you're going to reduce your risk down to up to 99%. Of all those spores are going to be gone. But it's something that we just don't do, and we know of it. Same with then soil movement. I just put a big X over this. This is tilling your equipment, right? You're not breaking up patches of clubroot. You're spreading it across the entire field with something like this. So that's where the recommendation of not tilling your soil or working your soil, especially if you have clubroot, because now all of your neighbors and the rest of your fields are going to have clubroot as well. This is a perfect way for this pathogen to take off. So that's just a huge no-no. Weed control. It's one of our ones that falls low on the list. We mention it because it's extremely important to be looking for clubroot symptoms in those off years. It is a brassica disease, so you are going to see it on some of our weeds that we are controlling. Like stinkweed would be an example of that. Dan, I know you got a few other ones on the list there. Wild mustard.
And flicksweed.
Flicksweed, yeah. So the real key piece here is just controlling your weeds. That's a given, right? We want to keep weeds down. But it doesn't take a lot of, early in the season, those plants will be susceptible. So they're, say, upwards of 3 weeks of growth, you could potentially getting galls forming on those plants. I know we struggle here controlling our volunteer canola in our soybeans and things like that. That's another reason there to control those plants. Anytime that there's a susceptible host in a field, you are running that risk of galls forming and keeping that life cycle going, adding more spore back to your soil. Patch management. This is something that's kind of new to us in Western Canada. We're still figuring the ins and the outs to it. It's complex. We're learning more and more each year. We've got a bunch of different work. This is just an example of what would be going on. We've run different cases in Alberta where we've got sanitation zones where producers are changing entrance ways to move away from those clubroot hot zones. We're starting to throw in patches where we potentially be liming or sowing down to some other non-susceptible host. So it's just something that we are looking at. They've looked at then pulling up plants in that infected region and burning them just to get rid of them and then sowing it down to some other grassy species and just staying out of that zone. No equipment through it, no movement, no transferring that soil, just staying out of that patch. And we've done this actually up in the Swan River Valley, the few cases there. It allows for that the spores to break down within that area, and hopefully then over time you can start growing and using that within crop production again. But it's staying out of it for a few years anyways. Even in those wheat years, you stay out of that patch, right? Because the spores are still there and they're still present within the soil. So we've mentioned our kind of our 6 silver bullets and how they don't stand up alone, right? They will fail, and that's why we need to be looking at this overall management approach. This thing is going to play maybe. All right, we've got our silver bullets and they're going right through. We can't just rely on crop rotation alone. We can't just rely on resistant varieties because we are starting to see failure of these, right? We need to be using them all together as an overall integrated approach.
In Alberta, the successful farmers that are farming in the heart of Clubroot, like I have some close friends that farm and are completely surrounded by Clubroot and by farms that are having trouble growing canola, even the Clubroot is so bad. And they've just by default been kind of doing these steps. One of them is a part owner in the alfalfa plant, so he grows lots of alfalfa. Another guy has a really good malt barley contract, so he grows malt barley. But they've been on a 3 or 4 year rotation for 15 years. They own their own equipment. They don't have custom equipment all over their farm. They kind of followed these rules by default, and they honestly will tell you that they don't know where the Clubroot is. They have it for sure. They have Clubroot on their farm, but they wouldn't know where it is because the spore loads are low enough that they're able to manage it. And I often hear people say that, you know, I'm doing everything I can, but my neighbor isn't. And I don't think your neighbor could possibly impact you enough to cause a problem if you're following the management practices. Sometimes we have to think outside the box, and we, for example, if you share a water run with your neighbor, you shouldn't be tilling through that water run all over the place. Our guys are grassing that water run in. It doesn't produce a crop very well anyway, and they're staying out of it so that they're not spreading the Clubroot more. But I do believe that this control strategy and recipe that we have is working really well in Alberta, and there's no reason why it wouldn't work here. And the thing is, we don't have the opportunity to go back in time in Alberta and start over, whereas you guys are kind of at that point where I think you can start to manage this like now. Yeah. And it's not easy to switch from a 2-year crop rotation to a 3. We have farmers all over Alberta that will come and speak to you about how long that takes, and we kind of think it takes about 5 years. You can't just switch your whole farm upside down next year and grow crops that you don't know how to grow or that don't make you money. So it takes time. So if you're on a 2-year rotation right now, I suggest that you look at ways to get to that 3- and 4-year rotation inside the next 4 or 5 years, which would include some of these tips that Justine mentioned.
Yeah. So, right, we've gone through each of the bullets alone and how they're not stand to hold, and that's why we need to take this shotgun approach, this integrated pest management approach. If we can throw all of these tools, we'll be able to manage it. And Dan highlighted perfect situation in that hot zone of Alberta, how we're able to do that. If we can incorporate all of these, we will be able to manage Clubroot here in Manitoba. We do have the step above because we've got some lower spore loads to work with. We've been fairly proactive with our scouting and our monitoring of this pathogen here in the province. So this is where that integrated pest management, using all of those tools together, will help manage it. So the recipe was back one. Hopefully you got a picture of that slide to begin with so you can remember exactly what we need to be doing to manage the disease. This has got to be the most frustrating clicker. PSI lab. Oh. I just wanted to mention the PSI lab. Here in Manitoba, the Manitoba canola growers are offering for each member a free Clubroot test, so a free soil test. This is just a good indicator of yes or no, you may have an issue. Dan alluded to the issues around soil sampling, but this is something to definitely take that opportunity and use. As a producer, each year you get a free soil sample, a free blackleg race ID test, and a glyphosate resistant kochia test. So meet us up at the booth at the Manitoba Canola Growers. We'll be able to talk to you about that program and definitely take advantage of that service to understand what's going on with some of your pests. For the Saskatchewan growers in the audience, the monitoring has changed there. So they now have to get permission to access your land. So if you want to get a free soil test, this is a way to do it. Sign up for the provincial survey. And so we've got a bunch of resources online. Clubroot.ca is kind of the housing body of all things Clubroot. If you're looking for the management practices we talked about today, if you're looking for videos, extension resources, new research, it's all up to date and on there. And we work with the grower groups on getting that information out in a bunch of different venues. You'll see some of the Clubroot videos are done that are available, and then some of the publications as well. So do make use of those resources. And with that, I think we're wrapping up.
Yeah,
2 minutes. Woo.
We'll be at the booth
Yeah.
this afternoon, I think from 1 to 5. I'll be hanging out there.
Yeah, Dan's there. I'll be there Thursday. But yes, if you are looking for the Manitoba Canola Growers booth, it's just up around the corner here along with Canola Council. We have a joint booth together. If you have any questions at all, we've got samples. We do have one Clubroot on stage here, but there's a bunch up at the booth as well if you haven't seen it before. So yeah, feel free to swing by the booth and ask us questions. And thank you very much for attending this morning's session at Ag Days.
Thank you.